Leachables are components that are migrating into my drug solution. Components are the packaging material.
Which are migrating into your drug solution. So now tell me, when is it going to be a problem? When it is in your packaging material, or when it migrates into your drug solution and then you get to know, okay, how it's interacting with all the components? What is important to study?
When it has been migrated into drug solution.
Did you get your answer? That is why we study leachables on the drug solution, on the drug product, not solution, any drug product. के जो कंटेनर में से लीच आउट होके तुम्हारे ड्रग फॉर्मुलेशन में आ गया है, okay, how is that interacting with your formulation? Is it causing degradation or this or that or whatever? All right, that is what we are studying in leachables. Now you will ask, but what about those things which are not leaching out? Even that can have some impact, right? So those are extractables, and that is why you study extractables. You assess extractables by assessing the containers.
Yes, ma'am.
Got it? So that is the difference between extractable and leachables. Leachables को बोलते ही leachable है because it leaches out into your formulation. And extractable को तुम forcefully extract करते हो just to study. Got it? दोनों तुम्हारे container में होता है, but one has a property to leach out. And where will it leach out? In your formulation. That is why you will study your formulation. Another one won't leach out. It will stay in your container. That is why you will assess the container. Samjha? Any doubts with this?
No, ma'am.
Good. Anyone else? Any other doubt? No doubt.
Ma'am, question number 7 from assignment.
We are going to discuss assignment.
Okay.
All right. We'll discuss it at the end. Besides that, abhi tak jo bhi sikhaya, parenterals is a little heavy topic, I know. So any doubts in that? So should we proceed? So today what we are going to do is, we are going to complete the topic. Processing बाकी है, right? तो processing का part complete कर देते हैं. उसके बाद MCQs भी थोड़े pending है, right or wrong? Pending. So you all need to tell me कि कौन सा कहाँ से MCQ pending है. I lost the track. Okay. So see, you all have the booklets with you all, correct? Huh? Yes or no? Yeah. Fine. So just tell me kahan se MCQ chalu karna hai. So we'll complete the topic, do the MCQs, and then get on the assignment questions. Fine. All right. So let's begin. Let me know if you can see the screen. दिख रहा है?
Yes,
Okay. Good. Fine. So yeah, the last part of this chapter is knowing. Abhi हमने सब पढ़ लिया. What are injectables? What are parenterals? What are the different types? What are the classifications? Acha, wait. I want someone to gist me up. Gist up to the entire class. हमने क्या क्या पढ़ा है? That would be better. Hello. If I ask a question, everybody goes mute and dumb and.
Ma'am, initially we
Yes,
started with the introduction, the definition of parenterals, that what are parenteral products. It also includes implants and other injectables. It also includes its basic characteristics, that it should be inert, free from pyrogen.
Just a minute. तुम बोलते जाओ. Accordingly, I will scan the slides. So it will be a quick revision for all of you. All right. Just hold on, Chinmay. And I want someone else also to take up. Chinmay will take up the first section. Okay, then I want someone else also to take up. Okay, Chinmay, shuru ho jao. So we started with what is it?
Parenteral route of administration. It includes implants and injectables. Then later we study the characteristics that all the products should be sterile, free from pyrogen. Then we studied regarding the pyrogens, that what exactly the pyrogens are. They are means lipopolysaccharide components exuded from the microorganisms. Then we studied about the disadvantages also, that it might cause the patient incompliance. Then later we studied regarding
These
the
are the
routes of
disadvantages.
administration.
Okay. Disadvantages इतना सारा पढ़ लिया, उसके बाद advantages भी पढ़े, correct हमने?
Yes मैम, एडवांटेजेस में मतलब बायोअवेलेबिलिटी अच्छा है, then rapid therapeutic effect, those are advantages. Further, the route of administration, that is IV, through skin, CNS, various others, many more route of administration. Within IV also there are more intramuscular, subcutaneous. Then later,
Hold on, Chinmay. Hold on. Can anyone else take up from here? Very good, Chinmay. It's quite impressive. Anyone else?
Ma'am, so
Who's
we have
this?
started with the route of
Chaitanya.
parental administration.
Hey, listen. तुम लोग जब बोलते हो ना नाम बोलो. I can't see who's speaking and all. Okay, so this
Am I
is
audible?
Chaitanya, right?
Yes.
Yeah, Chaitanya.
So
Chinmay,
we have
good
studied
job.
we have studied the various route of parental administration. It may be intradermal, subcutaneous, in the CNS, intraarterial, intracardiac, and so on. Then further, we have done with the classification and all that part of the parenterals. What actually what is actual classification depending on their volume, like small volume parenterals, large volume parenterals. Also, we have studied their overall formulation part of that of particular parenterals, and what is the specific volume required for the large as well as small volume.
Hold on. बहुत jump मार दिया. We studied the types of form. After the routes of administration, ये types of formulations पढ़े. ये parenteral में we have solution, suspension, emulsions, right? Reconstitution, all of that. उसके बाद pharmacopoeial classification किया. Yeah, this is what you spoke about. Small volume, large volume, lyophilization.
Yeah, that's the means.
बोलो lyophilization में क्या किया?
There we have started seeing what the actually lyophilization process is, and means actual freeze drying, how it takes place, and what are its advantages, disadvantages. Further, also we have done kind of actual how the process takes place overall, lyophilization cycle, how the overall process takes place and all that. And then further the various components of parenterals, like we use preservatives, antioxidant, tonicity modifiers, various buffers, and depending on the formulation, the solvent also varies what we use. Also, we use 4 solvents because somewhat if the drug is not soluble in that particular solvent, so to enhance its solubility, we use particular solvent. Also, the buffers we have studied, that is phosphate buffer, citrate buffer, and so on. Further, chemical stabilizer, additives, also the various surfactants like UNAT, all that, so that they comes under the excipients category that we use.
Right. So these are the different things we studied, right?
Yeah. Yes. And
Very good.
genetic agent also like EDTA and EDTA we studied.
Yeah, correct. Very good, Chaitanya. Very good. Now, after that, what did we study?
containers and closures.
Yeah, fine.
In,
Go ahead.
in that, we defined what is a drug solution and what is a drug product. We defined what is secondary packaging, tertiary packaging, what are the different kinds of tests we perform, that is the leak test, and we define the stability conditions and the different packaging materials, that is containers and closures, which is consisting of glass and plastic as container, and closures is rubber seals and aluminum seals, rubber stopper and aluminum seals. In that we studied ampoules, which can be made up of glass or plastic, vials, which are made up of only glass, and the different capacities of ampule, that is 1 to 10 ml, vials up to 500 ml, then cartridges, syringes, bottles, and infusion bags. And then we studied about glass in detail, that it is silicon oxide tetrahedron, it's for small volume parenterals, and there is flaking of glass. We have
Then
different
type.
types of glass. Type 1, 2, 3, 4. Type 1 is borosilicate glass, which is for injectables. Type 2 is soda lime treated. Type 3 is soda lime glass. And type 4 is for non-parenteral. And for neutralizing the internal surface of type 3, we use SO2 at 500 degrees Celsius. And that is why type 3 can be used for injectables, but only for single time use, not for multi-use.
Then different tests.
And then we study the different kinds of tests. That is, powdered glass test is only for type 1, 3, and 4. And for type 2, we have specifically water attack test. And then we studied the disadvantages of glass. That it's the different disadvantages.
And then plastics and rubber closures and all of that. All right. Okay, very good, Yogesh. Then next class, what did we study?
Regarding the tonicity, which should be
Someone else. Chinmay ho gaya, Chaitanya ho gaya, Yogesh. Who's the next person who would like to just give an overview? And you have slides to help you. Who will go ahead? Tanika, Malika, Rutika. So no one for tonicity and tonifiers. You all will have you have slides for your reference. Come on. Okay, fine. You only go ahead, Chaitanya.
So we have studied the tonicity modifiers. What is actual purpose of the tonicity modifiers in parenteral formulation? We studied the concept of tonicity. We studied the passive transport text, which basically a study about the osmosis, what is it, the transport of the solute from the higher concentration to low concentration. Further, we have studied there is isotonic solution, hypertonic, hypotonic, and we have studied that in hypertonic, the osmotic pressure is more than plasma pressure. Where we have studied the RBC gets shriveled in the hypertonic solution. Then we also studied isotonic, where the net osmotic pressure remains same. Further, the water concentration inside and outside the cell also remains the same in isotonic solution. Further, hypotonic, we have studied where the solute concentration is low. And also and also can undergo hemolysis, and also the cell can become turgid in hypotonic solution.
Correct.
Then further,
Hemolysis hoga in hypotonic solutions.
Hypotonic. Then for the tonicity modifiers, we have studied what is important in parenterals. Then uh, we have studied the difference between tonicity and osmoticity. What is actual difference in that? We have studied that the 2% solution of boric acid is iso-osmotic with blood. That example we studied. Then further we studied the isotonicity calculation various methods. The first is freezing point depression method. Then further we have divided into then class 1, class 2,
Yeah.
that part.
This is same, huh? Basically freezing point depression, cryoscopic.
Right. Then we have studied the sodium chloride equivalent method, then the white Wilson method, and then Sproul's method. And we have
Okay.
seen the respective formulas according to that, and we have done with some numericals with regards to them. Yes.
Okay, very good. All right. Good job, Chinmay, Chaitanya, and Yogesh. I hope, like, I'm so sure that you all have understood the things. I hope even others have. All right. So now let us conclude with understanding the final process. Sab kuch dal diya abhi. Let us see how the production is done and how it actually comes in the vial. All right. So we'll go about with the production procedure, sterilization, and where is it prepared? How is it prepared? All right. So production procedure में सबसे पहला तो obviously आएगा cleaning the containers, closures, and equipments, because तुम्हें जो vendors के पास से मिलेगा, that will obviously not be clean and all. It must be made clean from dirt, debris, any chemical residue. All right. So क्या होता है ना इंडस्ट्री में ऐसे एक जेट लाइंस जैसी होती है. So वायल्स तुम्हारे होंगे कन्वेयर बेल्ट में एक लाइन के साथ, एंड ऐसा एक जेट स्प्रे जैसा होगा, वेरी फाइन नोजल. ऑलराइट. और इधर तुम्हारा वायल्स आएगा. फाइन. एंड बहुत फोर्स के साथ इट विल फ्लश इन वाटर. It will flush in water to remove all the dirt and debris and all. And then it can be autoclaved also to make it pre-sterilized, or it will be simply dried in a hot air oven. All right. Separate equipments have to be used for oncological and biological products. This is very important. Production, ye cheez gaant ban lo. Production house mein, okay, suppose if a company is making your normal products, okay, if it's making onco products, and if it's making a biological product. Biological matlab tumhara vaccines ho gaya, tumhare antibodies, and everything what comes under your immunomodulators and all of that. Onco products and even immunomodulators product, like your sirolimus, tacrolimus, okay, all of those things, and these are others. So if a company is having a capacity for all these four, if it's having all these four types of products, those should have different plants, different manufacturing units. Tum paracetamol wale manufacturing plant mein cisplatin and your pharmacistins and your onco products tum develop nahi kar sakte. ना ही तुम्हारा immunomodulators, ना ही तुम्हारा biological. So that is what is being said here. Separate dedicated equipments for onco and biological products. Also immunomodulators. It is a rule in the GMP guidelines as well, because these three are considered as very hazardous. तुम्हारे onco products जो होते हैं, oncological products, what are they? What do you mean by oncological products?
Cancer drugs.
Anti-cancer drugs.
Chemotherapy.
Yeah. Correct. Chemotherapy, anti-cancer drugs. So your anti-cancer drugs are so potent कि वो तुम्हारे normal cells को भी मार सकता है. Correct. That is why it's considered as hazardous. Immunomodulators, they can modulate अच्छी खासी immunity also, right? It's used to overcome some diseases, but to a normal person also it can cause issues. So that is what is being said here. And even your biologics. Fine. So that is why unke plants bhi alag hote hai. Everything for it is different and dedicated. Fine. So then after you are done with your cleaning of your containers, closures, and equipments. So equipments ke cleaning mein you have something like this, CIPs and SIPs. Okay, ये मैं अभी भी इसकी बात कर रही हूँ, cleaning की बात कर रही हूँ, CIPs and SIPs. तो CIP is called, उसका full form is cleaning in place, and SIP is sterilization in place. All right. So cleaning in place matlab when you have huge tanks and something which cannot be disassembled or which cannot be moved, how are you supposed to clean it? You're going to clean it at its place. बड़े tanks में लोग अंदर उतरते हैं tank के and बड़े pipes and all के साथ they will clean it with water hoses. Okay, water hoses के साथ उसको clean किया जाता है. And sterilization in place is also the same things. जगह पे ही sterilize करते हैं by using steam and all of that. Fine. So that is and it's done usually by VHP, vaporized hydrogen peroxide. Hydrogen peroxide. Fine. These are industrial terms, CIP, SIP, and all of that. Okay. So for huge containers and equipments which cannot be disassembled, वो सब के लिए ये techniques use की जाती है. Then is product preparation, obviously. So based on the formulation, agar wo suspension hai, emulsion hai, jo bhi hai, and maintaining all the requirements, tum apna product prepare karoge. Now, there are some products which are made normally at room temperature. There are some products, especially in your oncological drugs, your chemotherapeutic drugs are very nasty. Very nasty. Okay. उनका preparation भी तुम्हें 2 to 8 पे करना होता है. 2 to 8 degrees Celsius. So what do you use? You use a jacketed vessel जिसमें ठंडा पानी घूमता रहता है, and in those conditions you will make your product. Fine. So the product preparation, what type of product you're preparing, what are its stability conditions required, what kind of sterility. Is some products जिसका terminal sterilization नहीं हो सकता है, तो उसको हम कैसे sterility maintain करेंगे?
Aseptic area.
Very good, Sejal. By aseptic processing, मतलब first step से ही तुम aseptic environment रखोगे. Fine. So your product preparation and all of that depends on what kind of product you're making and उसका do's and don'ts क्या है. समझा? Then comes filtration. If say aseptic processing की जरूरत नहीं है, TS मतलब terminal sterilization का है बात, then how will you do filtration? Just a minute. Okay, so filtration matlab terminal sterilization. So which are the different filtration techniques? Can you all hear me? Okay, so next step is filtration. Now let us understand the science of filtration. There are dedicated departments and dedicated industries which work on the science of filtration. Can you all hear me? Am I audible?
Yes, ma'am.
Ma'am.
Yeah.
Yes.
So there are filtration itself is a different science. You have companies like Millipore Wing of Merck, which specially deals with filtration. There are different types of filters available, different sizes of filter available. Pore size भी different होता है. Filter membrane का भी size different होता है. You will have a small filter membrane like 13 mm. You will have 25 mm का diameter होगा. You will have 75 mm का diameter होगा. You will have different MOCs का होगा, material of construction. Okay, you have PVDF filters, you have nylon filters, you have PTFE filters. It's PTFE or PTFE filters. Then you have PES filter, some polyethylene styrene, something like that. Okay. You have different types of filters. And some of these filters are hydrophilic, some of them are hydrophobic. I think nylon is hydrophilic, PVDF is hydrophobic. So depending on what you want to filter and what is the nature of that, you will select the filter. Kitna bada filtration unit hai tumhara, uske hisab se you will select the size of filter. You have something called as syringe filters also, jo tumhari syringe pe tum attach kar sakte ho for some analysis and all of that. Okay, so filtration science is a vast science altogether. Okay? And filtration when used for sterilization purposes is called as cold sterilization, because tum heat apply nahi kar. Correct? Jaise autoclaving mein ya dry heat sterilization mein, there is heat energy you are putting in, right? Isme kuch nahi hai. So this becomes very important for your thermolabile type of drugs. Okay? Yeah, this is what I was talking about. PVDF, PTFE. This is PTFE. PTFE, your Teflon filters. Then nylon N99 वैसा होता है. Okay, so PTFE, nylon, PVDF, PES filters. Okay, these are commonly available, commonly used filters. तो ये जो बैक्टीरिया वगैरह निकालने के लिए, whenever you are saying you are using a process of cold sterilization, कितना माइक्रॉन्स का होना चाहिए उसका फिल्ट्रेशन मेम्ब्रेन? It should be 0.22 माइक्रॉन. जब तुम 0.22 माइक्रॉन से फिल्टर करोगे, ओके, जब तुम 0.22 माइक्रॉन से फिल्टर करोगे, then only it is qualified as a sterile product. Okay, so cold filtration और sterile filtration मतलब 0.22 माइक्रॉन के फिल्टर से करना है. This 0.22 माइक्रॉन is the pore size. हाँ, फिल्टर में जो तुम्हारे पोर साइज होते हैं वो 0.22 माइक्रॉन का होगा. If you do it from 0.45 micron, it's not considered as sterile filtration or sterilization. It's just considered as filtration. So this is very helpful for your drugs which are thermolabile. Okay, but it becomes challenging for your suspensions or nanoparticles and all of that, because 0.22 micron मतलब कितना नैनोमीटर हुआ? 1 micron is how much nanometer? Tell me that. Oh my God, you all should know this. 1 micron is how much nanometer?
10 to the power 3.
हाँ, मतलब 1000 नैनोमीटर.
Yes.
तो 0.22 माइक्रोन कितना हुआ?
220 nanometers.
Thank you.
20 nanometers.
Thank you. Okay, so इसका पोर साइज स्टेराइल फिल्ट्रेशन का is 220 नैनोमीटर्स. अभी तुम्हारे नैनो मटेरियल, नैनो पार्टिकल्स या लाइपोसोम्स या some suspensions in that case, what if they are 300 nanometer or 400 nanometer? तो फिल्टर हो पाएगा? नहीं, फिल्टर ही क्लॉग हो जाएगा. You will lose your product. So in that case it becomes challenging. So for that, your formulation which you are making must be less than 220 nanometer. Got it? It must be less than 220 nanometer. अगर नहीं है, like for example your microspheres. Okay, microspheres I don't think market में है, is just in research right now. So microspheres जो है, they aren't, you cannot filter them through 0.22 micron because उसका size ही 1 micron से या 5 micron से चालू होता है. Fine, in that case you cannot sterile filter them, because वो choke हो जाएगा. Are you all understanding that? So these are the things. So now filtration are used for two purposes. Filtration is used for sterilization also, and filtration is used for clarification of your product also. Suppose you are going to autoclave it. But जब भी तुम injectable बनाते हो, it should be free from particulate matter, free from fibers and all of that, right? So make a note that filtration is used for two purposes. First is clarification. Clarification, माने के to get a clear product, and second is for sterilization. So filtration करना ही करना है. It is a mandatory step. Fine. But क्यों करने वाले हो, that will depend. So the functions, yeah, fine. Membrane filters are commonly used. Commonly used नहीं, वही use किया जाता है. Membrane filters ही use किया जाता है. And these are the names. Please remember these names. Now filters का भी test किया जाता है. You know, when in industry you are developing a process, you have to see कितने rate पे filtration हो रहा है, speed पे, then कितने pressure पे हो रहा है, because एक जगह से you will apply vacuum. कौन सा pump use करने वाले हो तुम? Are you using a reciprocating pump for filtration, a peristaltic pump, or you know, different, or a vacuum pump? ठीक है, ये सब. So these are some important tests for filtration. First is the bubble point test. Bubble point test. Bubble point test क्या बोलेगा? The integrity of your filter. कितना pressure वो maximum tolerate कर सकता है? That is your bubble point test. Okay, it's also called as filter integrity test. Second is pressure hold test. Okay, then is diffusion or forward flow test, how the flow is. Then is microbial retention test. Microbial retention test. तो उसमें क्या करते हैं? एक microbial media लेते हैं, उसको pass करते हैं. So ideally क्या होना चाहिए? All your microbes should be clogged, or all the microbes should retain on your filters, correct? Filtrate should be pura sterile. So once they pass, okay, this is a testing. This is one type of test, microbial testing. तो पूरा filter microbial culture pass किया, microbial solution pass किया. Then filtrate को test करेंगे. Filtrate should have 0 microbes, and your filter should have all the microbes. Okay, so that is a microbial retention test. Is it clear, everyone, about filtration?
And
हाँ?
yes, मैम, जैसे आपने बताया कि क्लॉग नहीं होना चाहिए आपका फिल्टर जो भी आप यूज कर रहे हो. तो
हाँ.
मैम जो पार्टिकल्स का साइज इतना छोटा है, तो यहाँ पे मैम एक इलेक्ट्रोस्टैटिक अट्रैक्शन वाला पॉइंट है. तो दोनों जो भी पार्टिकल्स एक दूसरे से इंटरेक्ट करके भी मतलब क्लॉग कर ही सकते हैं ना फिल्ट्रेशन के फ्लोर को.
So in that case, no, you have serial filtration also. Okay, now this becomes very specific. Serial filtration माने के, I'll tell you, this is one thing which I faced also with one of my product. मेरा एक product ना filter ही नहीं हो रहा था 0.22 micron से. Even though उसमें उतना कुछ था नहीं कि clog हो जाए. So what I did was I first passed it through 0.45 microns. 0.45 माइक्रोन से एक बार पास किया तो जो भी बड़े पार्टिकल्स है वो उधर ही निकल गए. एंड देन आई डिड स्टेराइल फिल्ट्रेशन. सो वैसे सीरियल फिल्ट्रेशन भी होता है, सीरियल फिल्ट्रेशन और सीरीज फिल्ट्रेशन वैसे करके भी होता है. गॉट इट? सो दोज आर द थिंग्स. इवन फॉर द पर्पस ऑफ क्लैरिफिकेशन, this was another situation which I faced. One of my buffer which I was using, it had some microbial growth. And I couldn't prepare a new one because it's very expensive. Okay, so what I had to filter it. So I filtered it through 0.22. It took 1 hour. Nothing was coming down. Hardly anything was coming down. Then I was like, in order to remove those microbes, let me first pass it through 0.45. 0.45 से एक दो बार पास किया, जो भी बड़े वाले fungal chains होंगे वो सब निकल गए. And then I passed it through 0.22, and then I got a clear solution. Literally it was visually different. एक पूरा turbid था and एक पूरा clear था, like water. So that is how also you approach filtration.
Okay, ma'am.
Okay. So ये filtration हो गया. And as I told you, filtration दो चीजों के लिए यूज किया जाता है. One is for clarification, which is mandatory, and another is for sterilization. Then is filling. Now filling can or cannot be aseptic. Fine. Aseptic filling, अगर तुम्हारा पूरा प्रोसेस aseptic है तो normal filling, if it's not aseptic, last में तुम sterilization कर. So in filling you have filling by volume, volumetric filling के हर एक vial में a particular amount of particular volume of ये जाएगा. Second is by weight filling. So तुम्हारे conveyor belt पे ही weight रहेगा, मतलब weight sensors रहेंगे and weight के हिसाब से dispense होगा. For viscous formulation weight वाला use किया जाता है. All right. And then is time pressure और time gravity dosing. मतलब पूरा पहले तो ऑप्टिमाइज कर लेंगे कि इसको ये फ्लो रेट से मैं भेजूंगी, मेरे ये सैंपल को इतने फ्लो रेट से मैं फिल करूंगी तो इतना क्वांटिटी फिल होगा, इतना फ्लो रेट इतना टाइम में इतना फिल हो जाएगा. ओके, सो यू विल टाइम योर इंजेक्टर्स. ओके, for 5 set of vials which are coming under conveyor belt, the fillers will come down. Okay, and it will stay for 2 minutes and 2 minutes में X quantity ही भरा जाएगा. Next 5 will come 2 minutes वैसे. So that is time pressure, time gravity dosing. Fine. After filling you have stoppering. First is stoppering and then is sealing. For ampules you have sealing. Sealing can either be bead sealing or bead sealing or pull sealing. Okay. And stoppering के बाद होता है तुम्हारा aluminium का crimping. Fine. In case of ampules, this is in case of ampules, you will have either tip sealing or pull sealing. In case of vials, first step will be stoppering. Next will be sealing or crimping. Crimping with your aluminium flip-off seal. Fine. And the last step would then be sterilization. Except for cold sterilization. Except for cold sterilization and aseptic processing. Except for these two things, your last step would be sterilization. Now let us look at sterilization. So these are the different techniques of sterilization. Let me also put aseptic processing. Aseptic processing is not a method of sterilization, but it is a way of obtaining a sterile product. Understand the difference. So yes, first you have autoclaving, which is very much preferred. Fine. उसको moist heat sterilization बोलते हैं, वरना steam sterilization भी बोलते हैं. It's taking place at 121 degrees Celsius. So इसमें कौन से materials use किए जाते हैं? Your heat stable, obviously. Glass and metals are compatible. Plastics को you cannot do. Obviously, they will melt. Fine. Then what is the mode of action? या फिर what's the mechanism? It coagulates the proteins of your bacteria and your microbes. Fine. Next is dry heat sterilization. That is by hot air oven. It's at 160 degrees Celsius. Again for glass, metals. Then for certain APIs also you sterilize your APIs, certain excipients, which are again heat stable. Okay. All of these, if heat stable, उसको अगर aseptic processing करना है, I told you, right? Aseptic processing में step 1 से everything should be sterile. All your components, all your equipments, all your excipients, your API, everything. In that case, तुम API and excipients, if heat stable, तुम dry heat से sterilize कर सकते. And even the mechanism of that is also coagulates protein. Basically, heating से your protein का coagulation हो जाता है, and that causes the death of the microbe. Then is filtration, which I told you, the pore size is 0.22, not 0.45, okay? Strictly 0.22 micron for filtration. Heat sensitive solution, okay, like amino acids, vitamins and all of that, biologics. prevents organism from passing through the filter, but does allow virus to pass. Virus can pass. Therefore, it's not a complete sterilization. That's the drawback. Then radiation में either UV या gamma rays use होते हैं. It causes DNA damage to its nucleic acid. But the problem here is it's effective only for surface. सिर्फ surface sterilization होता है. Okay, this is just for surface sterilization. But heat sensitive solids and plastics के लिए काम आता है. इसके लिए use किया जाता है. Again, it is important for aseptic processing to sterilize your raw material, your components. And then is gas sterilization. इसमें ethylene oxide and formaldehyde use करते हैं. इसको fumigation भी बोलते हैं. This is usually done to sterilize rooms. All right. Again, it becomes important for aseptic processing. This becomes important. This becomes important. Even this becomes important. All right. Mode of action is it inactivates enzymes for solids and some plastics. But पूरी की पूरी rooms को भी तुम formaldehyde या ethylene oxide से fumigate कर सकते हो, sterilize कर सकते हो. Fine. Now, how will we know that the sterilization technique which we are using is effective or not? We need some indicator, right? So what kind of indicators do we need? It depends on the sterilization technique. For example, filtration हुआ है कि नहीं effectively. How will we get to know? By using the biological indicator, which are nothing but microorganisms. In case of filtration, it's Brevindemonas diminuta. All right. So मैंने जो बोला ना microbial culture pass करते हैं, वो इस वाले microbe का culture pass करते हैं. Fine. Brevindemonas diminuta. उसका pass करेंगे and then test करेंगे how effective your filtration is. For moist heat sterilization and for your hydrogen peroxide or peracetic kind of sterilization, Bacillus stereothermophilus is used. For dry heat sterilization and for your fumigation or gas sterilization, Bacillus subtilis variety Niger is used. And for radiation, Bacillus pumilicis. Rough idea how to remember it. Filtration के लिए, filtration में कैसे होते हैं? Pores minute होते हैं, right? So you are going to use bacteria B. diminuta because your pores are minute. This is how you all can remember. For moist heat sterilization, and uska partner kaun hai? Hydrogen peroxide. You need some thermophilus bacteria, right? So you will use Bacillus stereothermophilus. And for dry heat sterilization, usi ka bhai Bacillus subtilis, variety Niger. And for radiation, you will use Bacillus pumilus. Understood? Did you all understand sterilization? Hello. Any doubts? Any doubts as of now?
Oh, man.
Okay. Now, if you want to do everything by aseptic processing, these are the different classes of rooms, the USP and the European Unions, okay? Classification based on US and European standards, all right? So European standards means grade A, B, C, D. US standards में it is class 100, class 1000, class 10,000, class 1 million, okay? Or 100,000. So what are its actual description? Class 100, जब मैं बोलती हूँ this is class 100 room. Class 100 room मतलब critical room, जहाँ पे तुम्हारा सारा aseptic processing होगा. This is class 100 room. And इसका मतलब क्या? There are not more than 3,500 particles having size equal to or greater than this. And there should be 0 particles, not more than 0 particles per meter cube of that room, which are of size equal to or more than 5 microns. Similarly, 100 में, if it's a clean room, again same, they're same, but EU में उन लोग ने 2 classifications दिया. So class 100 को critical या और clean room ही बोला जाता है. Fine. अभी class 10,000 is a controlled room. Fine. उसमें क्या होगा, और एक 0 बढ़ा दो, इधर 2 0 है, सॉरी 2 0 और बढ़ा दो. Fine. 3,50,000 and 2,000. इधर 4 zeros है, इधर again 5 zeros है, एक 0 बढ़ा दो. This is pharmaceutical room. This is for your normal OSDs and all of that. Class 100 and class 1000 also becomes very important. Fine. So yeah, this is the difference between class 100 and 1000. Actually, this is class 1000. Okay. So yeah, class 100, as I told, it's very important. So how do you maintain class 100 room? You maintain class 100 room by installing some kind of filters, which won't allow passage of 5 micron or 0.5 micron, these size ranges of particles. You need some filters which will control your air system. तो पहले तो एक air controlling system होना चाहिए, वो system को बोलते हैं HVAC या HVAC. Okay, HVAC या HVAC system बोलते हैं. What does it do? It supplies aseptic air, and it stands for HVAC or HVAC stands for heating, ventilating, and air conditioning. So you can control the HVAC system. Heating माने कि you can also control the relative humidity of a particular room. All right. Then वो वाले circulation में you need certain filters, and those filters are called as HEPA filters. Very important for parenteral manufacturing. HVAC and HEPA. HVAC system and HEPA filters. HEPA filters का full form क्या है? High efficiency particulate air. Okay, it's not written here. High efficiency particulate air. Fine. And this is the speed, 100 feet per minute का sweeping velocity होना चाहिए for the incoming air. So how are HEPA filters defined? It's an important definition. This slide itself is important. 99.99% Dettol के जैसे 99.99% or more efficient in removing from the air 0.3 micron का particles, which are generated by vaporization of Imori 3004. Fine. So ये जो Imori 3004 का जो पार्टिकल होता है, उसके वेपराइजेशन पे जो छोटू पार्टिकल्स निकलते हैं, वो पार्टिकल्स का 0.3 माइक्रॉन्स भी आना नहीं चाहिए. 5.5 माइक्रॉन का लिमिट है, बट वी आर ऑपरेटिंग एट 0.3 माइक्रॉन का लिमिट्स. That is what HEPA filter is. Fine. And laminar airflow units, they will help in controlling the air coming from these filters. So basically what you see is a LAF, laminar airflow units. You also have vertical airflow units. Laminar airflow मतलब सामने से आएगा LAFs, and WAFs मतलब vertical airflow units. So वो units पे you can see that वो unit के पीछे तुम्हारे HEPA filters होंगे. And how do you control that? You control it by HVAC systems. Fine. So class 100 area. And one more important thing here is that the class 100 area is maintained under positive pressure. ये समझा किसी को what is meant by this?
Ma'am, LAF and VF से बब समझाओ ना ma'am.
LAFs मतलब laminar air flows. It will come from sides. यो तुम्हारा जो air flowing होगा, सेंट्रलाइज्ड एयर कंडीशनिंग देखा है ना, वो वर्टिकल होता है. सो वैसे अगर ऊपर से आता है तो उसको वर्टिकल एयर फ्लो यूनिट्स बोलते हैं, साइड से आता है तो लैमिनार एयर फ्लो यूनिट्स. तुम लोगों ने you all might have seen, no, in movies and all of that, scientists are working in a hood kind of a thing. Hood के सामने बैठ के वो लोग पाइपेटिंग कर रहे हैं, cell cultures बना रहे हैं. So those hoods are having laminar airflow units. Okay, कि इधर से उधर पार्टिकल नहीं जाएगा, because air is coming सामने से. So कुछ भी पार्टिकल ऐसे नहीं जाएगा because of the air flowing in the opposite direction. So this is to avoid contamination. Now what I mean over here is that the class 100 rooms are maintained under positive pressure. Matlab, whenever you are entering these rooms, no, they are always having double door system. Ek door khola, you come in a chamber. Only when that first door is closed, you can open the second door. And then you enter your main area. So ye double door system hua. Now जो अंदर वाला room है, that is under positive pressure. मतलब the air pressure over there is a little higher than the air pressure in that बीच वाला chamber. And वो बीच वाले chamber का air pressure is slightly higher than the बाहर का surrounding. Okay. So say this is your lobby of your manufacturing unit. This is the door. This is the first door. This is that in between chamber जहाँ पे तुम्हारा lab coat वो सब होगा. Again, मतलब specified lab coat या जहाँ पे तुम वो protective PPE kits पहनोगे and all of that. This is one more door and this is your manufacturing area. Okay, this is your manufacturing area. Let me be precise. Your class 100 area. ठीक है? So यहाँ का pressure is going to be very high compared to this. ठीक है? So if the air pressure here is high, then अगर ये door open होगा, तो हवा अंदर से बाहर आएगा कि बाहर से अंदर आएगा?
अंदर से बाहर.
Correct, अंदर से बाहर आएगा. And when this door is open, तभी भी अंदर से बाहर जाएगा. This is to avoid contamination. Because अंदर वाला हवा is clean, it's sterile. वो बाहर जाएगा, that is okay. But बाहर का तो it's not clean, right? It's not class 100. So वो अंदर जाएगा, that is a problem. So this is what it is. Maintaining a positive pressure. कि अंदर का clean air can flow out. So even if there are cracks, no, in this room, even if there are cracks in this room, or if there's anything, atmospheric pressure से जब भी high होगा, तो अंदर का हवा ही बाहर जाएगा. This is to avoid contamination. समझा सबको?
Yes, ma'am.
Good. So this was about the processing, wherein we learned what are the processing techniques, what are the filtration techniques, sterilization techniques, and how do you maintain certain areas. So ये पूरे में से this one is a very important slide. This one and this one. These 3-4 are very important slides. All right. Fine. So I'm assuming you all don't have any doubts. Do you all have any doubts? Then abhi mujhe puch lo. Then we'll start MCQs.
No, ma'am.
Okay. So can we start with MCQs?
Yes, ma'am.
Yes, ma'am.
Why is Chinmay sounding so tired?
Ma'am, yesterday I was having fever.
Oh my God. How are you now?
Now better, ma'am.
Now better, man. Okay. कौन से MCQ पे we stopped? Tell me. Tell me. Huh? Question number 10. What is the question? Tell me, because मेरे इधर का numbering थोड़ा different है. Okay. Lyophilization was the last one. This one was the last one, principle of Lyo.
Yes, ma'am.
Okay, what is the principle of Lyo? Tell me.
Sublimation.
Sublimation.
Yes, thank you. Okay. What is not true for infusions? ये नहीं हुआ था. You all have your booklets open with you all?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay, yeah. So tell me, question 12. What is not true for infusions?
Infusion contain antimicrobial.
Okay. Infusion contain antimicrobials? No. Why no? Because they are LVPs. Correct? Because they are LVPs. That is why. So this one is correct. Infusions are LVPs. ये चीज तो correct है. Infusions can be emulsions?
No.
Ma'am, in case of TPN, infusions are emulsions.
Correct. In case of TPNs. They are oil in water emulsions, Chaitanya. I told you oil in water
Yes, yes.
emulsions can be given in LVP. Water in oil you cannot. Water in oil कौन से route से दिया जाता है? I
Iron.
या फिर SC से. Okay. Infusions are more than 500 ml. Even this is right. So the correct answer is A. Next. There is a not in the question, so pay attention.
A option.
E option, ma'am.
Correct. Because emulsifiers are component, containers are fine. So don't get confused. Okay, containers are not. They are components. API vehicles. So the correct answer is E. Select all correct options. There can be more than one correct answer. Okay, so be careful.
Option A and C option.
What about others? Come on, why am I not getting answers?
Ma'am, A and C.
No.
All of these,
Option?
all of these D option.
All of these is the correct answer. Aren't others sleeping? मुझे बस 3-4 लोगों का ही आवाज आ रहा है. चैतन्य, चिन्मय, योगेश, साहिल. What about others? You all might as well don't attend the class now. Just watch the recording. Are you all there? Sanika, Chaitali, Siddhi. Sujit, Tahir, Payal.
Yes, ma'am.
Then why are there no responses
Yes,
from you?
ma'am.
Yes,
Why are you
ma'am.
sitting like dumb? Payal, tell me what is the answer here? D is the answer. Explain why D is the answer.
because they all are correct. They are all come in correct pair.
Can you explain me that? Why are pyrogens water soluble? Tell me that.
Um,
You know, the more you interact, the more you'll retain things in your mind. What about Sujit? You're simply just logged in and you aren't bothered about what is going on in the class. What kind of answer is that? Because दूसरे options नहीं, सब सही है इसलिए that is the option. Give me an explanation. Sujit?
Yes, sir.
Why are pyrogens water soluble? Tell me.
Ma'am, pyrogens are made of lipopolysaccharide. The lipo part shows the pharmacological action, but the polysaccharide is water soluble.
Correct. That is why it makes them water soluble, and that is why you can't filter them. All right?
Yes, ma'am.
Thank you, Sujit. I'll be more glad if you khair abhi ye to aakhri class hai. Anyways. Payal, you need to pay more attention and don't give stupid answers like that. What is true for vehicle? Sanika. Sanika, I think she's not there in the class. Maithili, Devesh, Rishali.
Ma'am, water is mostly preferred.
Okay, Devesh has also put answer in the group, correct. What is true for vehicle?
Ma'am, option A, water is mostly preferred, but only in water or injection grade.
Okay. And?
and these also.
So all of them are right, actually. Water is mostly preferred, but only in WFI grade. See, here I'm not asking you all in the format of GPAT. I just want to test if you all have understood this or not. All right. So water is mostly preferred, but only WFI, correct? What about
Yes,
option B? Is it right or wrong? Tell me that.
Right. It's right now.
It's right. Who's this? Rishali.
Yes.
Yeah. Explain option B. Devesh will discuss that after this. Give me an explanation for option B. Someone. Vegetable oils are not preferred because they turn rancid. Is it true or false?
Ma'am, it is false.
It is false. Correct.
It is false.
They
Because
can be used, right?
Yeah.
We use arachis, matlab peanut oil. We use parenteral में और कौन सा होता है? Soybean होता है. Then cotton होता है. These all are used parenterally. Fine.
Linseed oil also we use.
Huh?
Linseed oil.
That's not used parenterally. I don't think so. Linseed oil is used for parenteral. Okay. Keep check on water miscible solvents can be toxic. Is it true or false? अरे यार एक क्वेश्चन पूछो मुझे 5 मिनट रुकना पड़ता है फॉर द आंसर. वाटर मिसिबल सॉल्वेंट्स टॉक्सिक होता है नहीं होता है? Tahir,
No, ma'am.
क्या no? Sahil,
Yes, ma'am.
C is
Water
true or
miscible.
false?
It is, ma'am. Water miscible cannot be toxic.
कौन से है water miscible solvents?
Alcohol, ether.
Ether is water miscible, huh? In which universe? Propylene glycol, glycerines, ethanol, they all are water miscible. Co-solvents की तरह use किया जाता है. But if they are used in excess quantity, it can be toxic. So this is also true. And oils cannot be used in some cases. Even this is true. For example, your IV infusions, like if you are making an water in oil emulsion, it cannot be used for IV purposes. All right. So the correct answer is A, C, and D. Agree? Anyone, any doubts? All right. I'll take that as a no. इसमें क्या question था देवेश? Can you unmute your mic? For previous MCQ, option A and B both will be right. All of them are right.
मैम एक्चुअली आपने लास्ट लेक्चर में ए और बी ऑप्शन बताया था सिर्फ.
A or B बताया था. Okay, fine. Let me confirm on this. I will drop you all the, with explanation, I'll drop you all a message. Okay? Maybe through this thing, the app. Fine? Shruti?
Yes, ma'am.
Okay. Question 16. Which of these help in improving solubility?
B option. All of the above. All of this. All.
Correct. Complexing of API with certain agent. What is the example here?
EDTA.
EDTA.
EDTA. Actually, here it should, you all should tell cyclodextrins. Okay. Even EDTA, but EDTA का application is not for increasing solubility. Cyclodextrin के साथ जो complex बनाता है, that is used for enhancing solubility. Buffers कैसे use करते हैं? कैसे काम में आते हैं?
They maintain pH at which
So for so pH dependent solubility में तुम्हारा buffers काम आएगा. Correct? Then water miscible solvents का कौन सा principle होता है? अभी just बोला था मैंने. Dash solvency. Co-solvency. And of course,
Solvency.
solubilizing agents. Yes. What about parenteral API? Which of these is true?
B option.
Correct. Only B is correct. Only B is correct. They cannot be manufactured in any equipment. They need to be manufactured in dedicated equipments. All right. And may have some chemical impurities. No, they must be totally sterile and pure. Okay. So B is the answer. Next.
C option.
C. Histidine.
Histidine is used for biology.
Like bovine serum albumin we have seen.
Hey, bovine serum albumin is not a buffer.
No, means that type of we have seen our explanation part. You were telling about that.
I didn't use bovine serum albumin for buffer and all. I used it for other purpose. Tell me for what did I use that? फंस गया अभी तू बोल के. Don't mute yourself now, Chaitanya. Tell me, what did I use bovine serum albumin for? What is it used for?
For biologics, we have seen in that regards about that.
You just remember it's used in biologics, but for what? It's used for coating of the glass containers, ताकि protein agglomerate ना हो. They're not used in buffers. Surfactant is used. Surfactant used in parenterals.
Option B.
Option
D.
D, based on its suitability. Correct. Which preservative you will prefer?
Option B.
Which one?
Phenyl mercuric benzoate.
Are you sure? Are you sure?
Ma'am, it might cause mercury toxicity.
Huh? It might cause? Mercury compounds will use for infants.
No, ma'am, it can't be used.
Acha, yeah, for the 14th one, the answer is A and B. Low molecular weight की वजह से you cannot do the reverse osmosis. Fine. So the answer for that is only A and B for 14th वाला. तुम लोग के MCQ booklet में I think वो 13 या 15 number पे होगा. Select the correct pair. Did you all make that change? Hello.
Yes, ma'am.
Yes. Now tell me, इसका answer is hexyl resorcinol. IM preparations for infants. This is used. Mercuric benzoates are not preferred. Okay. Antioxidant preferred for acidic parenteral formulation. For acidic.
And A option.
Correct. Metabisulfides are used for acidic preparations. What is true about pH and buffers used for parenterals? What is true for this?
Ma'am, I think A and B.
Devesh.
Ma'am, all are true.
Correct. All of these are true. Come on. I know this was a little difficult question. pH value should be 4 to 8 prior to administration. ये मैंने बोला है. Citric acid is used as a buffer as well as an acidifying agent. Okay. And phosphates is a buffer but not an alkalinizing agent. All right. So that is a thing. povidone is preferred over HPMC for IM injection. True or false? इसका भी मैंने अलग से बोला था. Last slide में था, I think.
True.
Suspending agents.
True.
Correct. So for parenterals, povidone, where is it? Povidone is preferred over IM injection. Fine. So with this, we complete parenterals. उसका MCQs. I think तुम्हारे MCQ booklets में भी इतना ही था, right?
Yes, ma'am.
Yeah.
Yes, ma'am.
So now let us just quickly discuss the numerical. Fine, we'll discuss the two numericals. किसमें डाउट है तुम लोगों को? Question 1 किसने पूछा था वो?
Ma'am, question number 7.
हाँ, question मतलब first question of the assignment.
Yeah, first question.
Just a minute. Fine. Tell me if you can see the slide. Okay. So this was the question, right? Hello.
Yes, ma'am.
This was the question. Explain me the question. Can someone do that? Yeah. Nobody understood the question.
Ma'am,
Okay.
I understood. Ma'am, I understood.
Okay, fine. Let me only say. Fine. So we have to prepare a 200 ml isotonic aqueous solution of this drug. Okay, thimerosal. इसका molecular weight दिया है. So पहले हम given में लिख देते हैं. Thimerosal का molecular weight we have. Fine. तो इसका molecular weight दिया है. Then the concentration जिसमें ये use करने वाले हैं is 0.2 gram in 1000 ml. 0.2 gram in 1000 ml मतलब how much in 100 ml? Point 2 in 1000 ml. तो 100 ml में कितना?
0.02.
0.02. So can I say उसका concentration is 0.02% weight by volume?
Yes.
Okay. And the L-iso, okay? L-iso for such compound is 3.4. So हमारे पास इसका पूरा कुंडली है thimerosal का. Fine. Wherein उसका molecular weight is how much? 404.8. I'm not writing the units. Then uska L-iso is what? 3.4 and concentration uska use jo kar rahe hai wo hai 0.02 percent weight by volume. Fine. Okay, so this is being used, L-iso for such a thing. Now we need to find iska sodium equivalent. Okay, we need to make it isotonic solution, but we cannot use sodium chloride. Okay, sodium chloride interacts with these mercury compounds. Thimerosal is a mercury compound, तो sodium chloride इसके साथ react करता है. So instead we need to use replace NaCl with propylene glycol, जिसका molecular weight इतना है and L-iso इतना है. So how much propylene glycol will you use to make it equivalent to thimerosal? Fine. ये पूछा है, मतलब how much dash amount, ये hint भी इधर दिया है, dash amount of propylene glycol is required to adjust इतना थाइमरोसाल का टू रेंडर आइसोटोनिसिटी. So first we'll find कि if we were to use NaCl तो NaCl कितना यूज करना है. And तुम्हारे क्वेश्चन में L-iso दिया है, so which method you will have to use? This is the hint. L-iso दिया हुआ है, so कौन सा मेथड यूज करेंगे हम? ऊपर लिखा है देखो,
Sodium chloride.
sodium chloride equivalent method you will use. Okay, we will use NaCl equivalent method. ठीक है, तो तुम्हारे फॉर्मुलेशन में थाइमरोसाल है प्लस देयर इज NaCl. Assuming we are putting NaCl, so isotonicity will be provided by NaCl also and will be provided by thimerosal also. अभी कितना isotonic होना चाहिए in presence of NaCl? 0.9% weight by volume of NaCl solution is isotonic with blood. Yes or no?
Yes, ma'am.
Yes,
So we will
ma'am.
find first thimerosal का कितना quantity is equivalent. Okay. So what is the formula? E equals to 17 into L-iso upon the molecular weight. Correct? So this is the formula, right? So 17 into what is the L-iso for thimerosal?
3.4.
0.4.
And molecular
3.2.
weight?
404.8.
Okay. So solve this. What is the E which you get?
0.14178.
14178. 0.143 around मिलेगा तुम्हें. Okay. Decimal सारे रखो. Use scientific calci. 0.143. हाँ, 143 और 14 something मिलेगा. Round it off to 0.143. So that is E. मतलब weight of NaCl to which drug is isotonic. Fine. So, and this is for how much? हमने कितना quantity of thimerosal यूज किया है?
0.02. हाँ.
0.04 gram in 200 ml. Correct? Correct?
Yes. Yes.
तो ये 1 ग्राम के हिसाब से है, तो 0.04 ग्राम के हिसाब से कैसे होगा? E equals to 0.04 gram into this. Correct?
Yes.
तो इसका आंसर क्या आता है?
0.0057.
Correct. 0.0057 gram. So this much of NaCl you need. Okay, you need this much of NaCl more. इतना additional NaCl तुम्हें चाहिए. Okay, because E, what are we finding? The weight of NaCl to which this will be isosmotic. Oh, sorry, isotonic equivalent. So now we know हमें 0.9% चाहिए total. Okay, solution का अगर concentration with NaCl 0.9% होता है, then it is equivalent. Correct? तो अभी 0.9% में से मतलब okay 0.9% in 100 ml. तो 200 ml में कितना चाहिए रहेगा हमें? Because we have to make 200 ml. Correct?
1.8 gram.
1.8 gram. अभी ये 200 ml में 1.8 gram we need things to make it isotonic. Fine. तो अभी हमने already thimerosal यूज कर रहे हैं, तो वो कितना contribute कर रहा है?
0.0057.
Correct. It is contributing 0.0057 gram equivalent to NaCl. It is using. तो बचा हुआ कितना NaCl हमें need? बचा हुआ कितना NaCl चाहिए हमें? 1.8 minus 0. 1.8 minus 0.0057 इतना बचा हुआ additional NaCl चाहिए, correct? तो ये कितना आता है?
1.79.
1.794. Okay, so you can make a conclusion कि इसका तो negligible है because almost ये 1.79 almost 1.8 जितना ही हो गया. Fine. So you need additional 1.794 gram of NaCl to make your 200 ml of this thing, thiomerosal equivalent. But now you cannot use NaCl. You have to use propylene glycol. तभी उसका सोडियम क्लोराइड इक्विवेलेंट निकालते हैं. तो व्हाट इज द फॉर्मूला अगेन? 17 into what is propylene glycol का L iso?
1.9.
1.9 and molecular weight
76.
76.09. So what is this?
0.424.
0.424. All right. So as per the formula, what's the formula? Additional kitna propylene glycol will you use, which is having sodium chloride equivalent, sorry, जिसको तुम्हें 1.794 से equivalent करना है. This is the weight of NaCl which you need, but you are not using NaCl, you are using something instead of it, जिसका equivalent कितना है? 0.424. तो उसका quantity कितना मिलेगा? So what is this? What are you getting? इसका answer?
4.21.
4.21
4.21.
yeah, 4.34 I'm getting. 4.3 I'm getting. All right. So much gram of propylene glycol you will use to adjust 200 ml of thimerosal instead of NaCl. So this is your answer. इतने लंबे sums नहीं आएंगे तुम्हारे GPAT में, but this is for you to understand the concept. Is it understood, everyone?
Yes, ma'am.
Okay.
Yes, ma'am.
Good.
Yes,
Good.
ma'am.
And I think the other two questions were simple, right?
Yes, ma'am.
Yes.
Yes, ma'am.
Great. So usme ka the next one was based on freezing point depression and other one was similar. Like that. Fine. So can we end the class?
Yes, ma'am. Yes, ma'am.
Okay. So I hope you all enjoyed studying with me and enjoyed studying Parenterals as well. Yeah.
Yes.
Yes.
All right
Yes,
then.
ma'am.
Okay, then good luck and contact me if you all have any doubts. And I don't know if we'll have any lecture ahead. I've been told this is the last lecture. So good luck. Bye. Take care. All the best.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Bye.
Thank you, ma'am.
Thank you all.